In 2020, a crack research lab was quarantined by a virus for a crime they didn't commit. These men and women promptly took all appropriate precautions before returning to the Washington University underground. Today, still wanted by other academic institutions, they survive as researchers of fortune. If you have a question in plant mechanobiology, if no one else can help, and if you can find them, maybe you can join the Haswell team. The dream of the Haswell lab is to understand how molecules, cells, and organisms sense and respond to mechanical forces. We want to know how cells maintain mechanical homeostasis, how they achieve mechanical signaling that leads to cellular decision making. We want to understand the structure of mechanosensitive ion channels, and we want to reveal the evolution of mechanosensory systems. So we've been working on these sort of 4 quadrants for about 13 years now, and we're starting to see a couple of themes. We're starting to uncover universal mechanisms of mechanoperception, ways of sensing mechanical force that spans across all kingdoms. At the same time, we're also starting to uncover ways in which mechanoperceptive mechanisms are tailored to the unique features of plant cells in particular, with their cell walls and their vacuoles. And finally, we're starting to reveal the emergent properties of channel populations and cell membranes. Now, these are some awesome goals, and accomplishing them would not be possible without the awesome lab group. Last year, we worked as a lab to put together a lab mission statement, which we take very seriously. It reads, in the Haswell lab, we ask big questions about plant biology and mechanoperception, and we answer them meticulously. We communicate our work honestly, clearly, and widely, and we strive to create a diverse community of support, growth, and fun. The challenges to studying mechanical signals and mechanotransduction include the fact that they are ubiquitous. So every molecular and cellular reaction, every developmental process, every stress response is to some degree affected by a mechanical signal, which makes it hard to remove that in order to study it. In addition, the mechanical environment and the mechanical properties of cells differs widely across kingdoms and even across different cell types within the same organism. So what you learn in one system might not apply to another. So perhaps as a result of the first two, the last challenge is that mechanobiology is understudied. The kinds of tools that we use to study nutrient perception or hormone signaling are really only just now being developed for studying mechanotransduction. So our approach to getting a handle on how plants sense and respond to force has been to focus on a class of proteins that, by their very nature, are involved in sensing and responding to mechanical forces. And these are mechanosensitive ion channels. So physical events from either the outside or inside the cell can change the physical properties of the plasma membrane, either by deforming it or stretching it. When the plasma membrane is deformed by force in this way, it can lead to changes in the geometry of any of the proteins that are embedded in it. So mechanosensitive ion channels are protein pores in the membrane that open and allow ions to pass across the membrane in response to increased lateral membrane tension, that is, tension that is in the plane of the membrane. I like to think of these proteins as translators, where they translate the language of physics, which is force, into the language of biology, which is ion flux. Okay, so let's talk just a little bit about what we're working on right now in the lab. There are 3 main research projects going on at the moment, and I'll talk a little bit about each of them. One is to study mechanical decision-making at the cellular level. This project is supported by the Center for Engineering Mechanobiology. A second project is to understand the evolution of mechanosensitive ion channels in the green lineage and beyond, and this is funded by HHMI and the Simons Foundation. And then finally, we have an NSF-funded project to try to develop the pollen grain as a model system for plant cell mechanobiology.
Hello everyone, I'm Ivan, a postdoc in the lab, and I will tell you a little bit about the model species we work in this lab. So we use several species which cover several of the major lineages of plant evolution. And first we have a flowering plant, Rhodopsis thaliana, which is perhaps one of the most studied plants in the world. It offers several great advantages. For starters, it's a very small plant, so it can be grown easily, as well as it grows really, really fast, which is a great advantage for a plant. And it's also a very powerful genetic model. Then we actually have a tobacco species. It's not a smoking tobacco, but it's called Nicotiana bentamiana. And besides looking pretty, it also is very useful for a transient transformation and protein expression. So you can actually get proteins within a day or two. It's very great. Then we actually switch and go to a completely different part of the plant phylogenetic tree and find this species, which is called Chlamydomonas ringhardi, and it's a representative of the green algae. This particular species is a single-cellular algae, and besides having a beautiful green color, it's also an extremely useful model for studying evolution of multicellularity, as well as several other processes. And then we go to another part of the tree where we now have another species. This is called the moss species, Viscometrium patens, and this is a representative of the lineage which is one of the first to conquer land, and hence evolved several adaptations to do that. This species has several body architectures, as you can see here. So this is the same species, even though they look quite different. It's just different growth stages, and they are extremely powerful genetic models for transformations that are really efficient, and also imaging of the system is really, really easy. So these are some of the species. These are the species we are currently working with. However, we are open to expanding and always introducing new model systems and asking more interesting questions, especially about evolution and evolution of the green lineages and how the process of mechanosensation has evolved within plants.
Hi, I'm Carrie Miller, and I'm a 4th-year graduate student, and I just wanted to briefly talk to you about some of the techniques that we use in the Haswell lab. So most of the time we're doing functional genetics, so trying to understand what a gene and its associated protein product is doing in the biological system. So in order to do that, we use a wide range of techniques. A lot of times we're using molecular biology techniques, so if you like doing PCR, we have that. We also do genetic screens, a decent amount of microscopy. We have a lab-owned confocal microscope that many lab members use. In the past, lab members have used patch clamp electrophysiology to understand ion channel function. I personally have done a decent amount of mathematical modeling. And so really the techniques that we use range quite a bit. And I think that's one of the strong points of the lab, is that we're not afraid to try a new technique in order to do better science and draw better conclusions. And so, we don't shy away from getting a new collaborator in order to do a technique that we don't currently have access to. So if you're interested in any of that, I recommend coming to say hi to the Haswell lab.
Hi, it's Jenny, here to tell you a little bit about our lab's culture. Our lab is really collaborative. Liz has managed to attract a group of people that are willing to drop whatever they're doing to help you with an experiment or an analysis. And all of us have a different area of expertise, and I found that everyone is willing to share that expertise with you. Our lab meetings are constructive, and everyone supports each other by going to each other's talks and editing each other's papers. Before the pandemic, we would support each other even outside of the lab. So personally, me and another grad student played in WashU's symphonic band, and a lot of the lab came out to support us. And I also do some volunteering teaching kids about science in one of the underserved neighborhoods in St. Louis. And I've just been so pleased that so many people in the lab have came out to support me and even teach some lessons of their own. I just wanted to say that when I joined WashU, I had no idea that I would join the Haswell lab, but once I got here, I was really attracted to the lab atmosphere and the sort of research I could do. And so I hope that you'll give us a try too.